Hello everyone. Today I'm going to teach you DNA sequencing. In this video, what is DNA sequencing and what are the method and what are the advances involved. Okay, so DNA sequencing is very important nowadays for personalized medicine, genetic diseases, clinical diagnosis, as well as the crime detection to DNA detective and kind of fingerprinting. So DNA sequencing in future it's very important technique. So in this video I will let you know how to do this one. Okay. So in this chapter, after this chapter, you can be able to tell the need of DNA sequencing and its importance, understand the basics of DNA sequencing, explain the advances in the DNA sequencing. Very simple, okay? But we need a very expensive machine, okay? Then, what is DNA sequencing? Actually, the DNA is made up of four different chemicals. In these four different chemicals arranged in a different pattern, so we need to get the pattern. Then therefore, if you get the DNA sequence, then you can manipulate. Just like a word in a written language is spelled by arranging specific letter in a certain sequence. Here the specific letter is the chemical. Guanine, adenine, thymine, guanine, thymine, something like that. Okay, and usually only the specific sequence, a gene has a characteristic order of arrangement of the nucleotide which comprise it. So it has a specific order, therefore it has a specific character, the gene. The purpose of DNA sequencing is to determine the order of bases in a DNA molecule. Okay, that's a purpose. So initially the classical mapping actually we don't get the DNA sequence, but we mutate the particular gene and we get the mutant. So we only mutate particular gene. That's a classic mapping. But recently, the RDNA technology that provided and genomic that provided many areas where we can make a specific sequence obtain. So we can make genomic library. Then we can construct a physical map of entire genome. Okay, it based on chemical characteristics of the DNA itself. Ultimately the entire genome is sequenced such that all genes are identified by both their location in the chromosome and their nucleotide sequence. So based on this we can find out the defective gene and important gene, so where that's what we can manipulate the gene. So actually, there is a Alan Maxim and Walter Gilbert. They invented one method, chemical degradation method of DNA sequencing. It is around 1977 they have discovered this technique and chemical degradation method, but now it's outdated. There are another method, Dr. Sanger. Dr. Sanger, he used some enzymatic method to detect the DNA sequence. Okay, so this is now very popular and it's also has been advanced, but the very basic is this one, Sanger enzymatic method, chain terminating method. Okay, so this method in this video I will explain. So Allen Maxim and Gilbert method, it's outdated but still it's important, but in this video we are not discussing this one, we are discussing Sanger's enzymatic method. Okay, so the elegant idea behind DNA sequencing. He's the Fred Sanger. He's very brilliant because he discovered this technique. It's very I don't know how he came up with this idea. Actually, if you see the DNA have a deoxynucleotide, but he made it. He produced some other than that other deoxynucleotide. That's a dideoxynucleotide which terminate the DNA replication. In this method, okay, so we call it a chain terminator. Okay, so how he done this one? Actually this technology is still in use. Okay, because this is a base. From this there are many advances nowadays. Okay, it's the technology changes quickly, but For many years we have used Sanger's cool trick, and he got Nobel Prize in 1980 and shared with Walter Gilbert and Bohlberg. Okay, so here how he discovered this method and how we can proceed. First, you need to know how the DNA replicate using deoxyribonucleotide and DNA polymerase. Actually, DNA is made up of four bases, right? And DNA replicates 5 prime to 3 prime. And when the replication start, okay, the new bases will add to this OH group. Okay, so the phosphate and OH group bind and phosphodiester bond. So in sequence the DNA will replicate by the action of DNA polymerase and deoxyribonucleotide. Okay, deoxyribonucleotide will bind by the DNA polymerase by the formation of phosphodiester bond. So we need OH group. We need OH group in the 3 prime end, then only the DNA polymerase add the new nucleotide. This is the basic. Okay, now what he did is Sanger, Dr. Sanger, what he made it, he made dideoxynucleotide. Dideoxynucleotide. Normally in the DNA replication, deoxynucleotide nucleotide triphosphate, where only 2 prime, in the 2 prime there is a deox, the oxygen is removed. Okay, this is a normal base. But he made it dideoxy where 2 prime and 3 prime the OH is, the oxygen is removed. So there is no OH group in this nucleotide. Therefore, here this wherever this nucleotide bind, dideoxynucleotide bind, there will be termination of the reaction. So there's no polymerization. Okay, so you have to remember this one: dideoxy nucleotide triphosphate and deoxynucleotide triphosphate. So how we can proceed? For example, this is a sample of DNA which we don't know the sequence. Okay, so now we are going to find out this the sequence by this method. So for this we need a small primer, and we need deoxynucleotide, and we need DNA polymerase. Okay, so we mix this all together and we divide it into 4 tubes. Okay, we put in 4 tubes, and each tube they will have dideoxy, specific dideoxy adenine, dideoxy guanine, dideoxy cytosine, dideoxythymine. Okay, so they all tube have the enzymes and deoxynucleotide. Okay, but in this we have separated 4 tubes. The 4 tubes, the dideoxy only one is available. Okay, so specific, the each tube have a specific dideoxynucleotide. Remember this one. So the polymerization occur, and this DNA polymerase add this deoxynucleotide as well as dideoxy. Wherever dideoxy added, then the DNA polymerase stop. Okay, the DNA replication stop. Then they will make a different size of band, different size of DNA fragment. Then after that, if you run the polyacrylamide gel, where we can run the DNA, where we can run the DNA, where it separate the DNA by the size. Okay, so this is adenine well, guanine well, cytosine well, and thymine well. So thymidine well. So we will add the reaction. After that, it make a different band. Okay, so we know that the smaller band, smaller DNA run faster than the bigger one. Okay, so now we have to find out the DNA replication start 5 prime to 3 prime. So finally, first we need to read the bottom, which is DA, and another one is that this one is T, and adenine, and thymine, cytosine, guanine. So this is a complement to template. This is not actual sequence, this is a complement. So from this we can derive the original template DNA sequence. For example, GC, CG, and TA. So this is the actual template sequence. This is a complement sequence we get from the gel. Okay, and sequence technology has undergone significant advancement since the discovery of the original technology, which is 1970. The Sanger discovered this one. And now that's this third generation technologies, gel-less. So we no need to run a polyacrylate gel. Instead of that, scientists added some fluorescent detection in the dideoxy molecule, dideoxynucleotide, where each dideoxynucleotide will produce different color. Based on that, we can easily find out which nucleotide is adding. So the each of the four nucleotide dideoxynucleotide fluoresces a different color when illuminated by a laser beam, and an automatic scanner provide printout of the sequence. Now it's a very simple beam. There's automatic scanner, there is a laser beam, there is a machine, it will give the sequence in a printout form. So capillary array electrophoresis, where so after the reaction, so you no need to run a gel. Okay, just this capillary electrophoresis, if you add the reaction, so the smaller fragment run faster than the longer fragment, and the dideoxynucleotide have the color. So each nucleotide have the color, so the laser beam will detect the specific sequence. Okay, and nowadays the next generation sequence, it is advancement of dideoxy, actually pyro sequencing, where they found out pyrophosphate release of pyrophosphate after the phosphodiester bond. And sequencing by synthesis is the Illumina, we add adapter, and this is very famous nowadays, Illumina sequencing. Sequence by ligation, there will be no need any DNA polymerase. Here we use lot of primer to attach. And another method is ion semiconductor sequencing, where the release of hydrogen ion, and therefore the changes in the pH will be measured. Accordingly, we can derive the DNA sequence. So Sanger sequencing is the method which prevailed from the 1980s until the mid of 2000. Now there's a second generation, third generation, now NGS. Okay, so a lot of advancement. So this advancement bringing the cost into very low. Okay, how the when in 10 years back, almost 20 years back, if you want to do DNA sequencing, it cost almost $100 million per genome. But now in 2011 it is reduced to $10,000 per genome. So nowadays it's reducing because of the advancement. And at the same time, the sequencing timing, it will come to within a days. Okay, sometime before it takes long time to sequence the DNA. Nowadays within a day we can complete a genome. Okay, sooner or later everyone will have their DNA sequence in your hand, and therefore this technology is very booming. And nowadays it comes under lower cost and less time. Thank you. If you have any question, and you can mail me. Thank you.